I'll start sharing my screen. I hope my audio is working well.
Yes, she got it.
Thank you. Okay, could you please confirm if this screen is now visible?
Yes.
Okay, thank you. Okay, I can see that participants have started joining. And we have around 4, 5 more minutes left before we start with the webinar for the program. And before we begin, it would be great if the participants can introduce themselves in the chat box. You can let us know where you're from and what is your background so that it will help us understand the interest and the background of the participants. Okay, then. So I guess it's time. We will start with the webinar without further delay. Okay, so hello everyone, a very warm greeting to one and all. I would like to welcome you all to the open house session on the industry-driven program that we are launching on next generation sequencing and multi-omic data analysis. So before we begin, let me take a brief moment to introduce you to the team behind the program. We are India and US-based bioinformatics and data science company, and we have had the wonderful opportunity to collaborate and partner with esteemed universities and industries across the globe. And as a part of our mission, our team is working with regional and international research mentors that are helping refine program logistics and adapting them to the needs of students and researchers around the world. Now, to meet the ever evolving demand of skilled bioinformaticians and data scientists, we have created innovative training and research offerings, including online courses, training programs, and workshops, research internship and fellowship, research services for faculties, scientists, as well as a dedicated degree program. Now, In the below, you can also see some of our lineup of online workshops that are upcoming in this month and in the next one. So if you're interested to explore any of these workshops, you can definitely reach out to us. And as I mentioned in my previous slide, one of the important offerings that we provide are the coursework. So, what do these coursework contain? They include textual and visual aids such as diagrams, infographic, and videos to simplify the complex concepts that are there in bioinformatics. There are also short tutorials and practical demonstrations that provide students hands on learning while helping them develop coding skills. Now, at the end of the coursework, students will earn digital certificates, which can be later added to their resume, LinkedIn profiles, digital portfolios, and so on. And all of these courses, they are fully automated and compiled into lessons in detail. And many times, students require additional resources such as videos of mentors that are going through these and explaining the concepts and doing the hands-on. So for such students, we have training programs and workshops which include a combination of lecture videos, expert tutorials, and guided lessons. And these training materials are designed to align with modern industry relevant curriculum, ensuring students are able to gain practical skills that are applicable in today's academic and professional settings. And we also have an online progress tracking system to help monitor students' progress and assist them wherever required. And this has two aspects, that is the workshop, which include live mentor guided sessions, and the training program, which comprises of recorded lecture videos. Now, here is a brief summary of some of the various open source tools and technologies that are covered throughout our program and courses. These are commonly used by researchers worldwide, and there are many more tools that we cannot cover through slides. But coming to the main program, that is the aspect of today's webinar. So this is a 4-month hands-on industry-aligned program that is combining wet lab basics with cutting-edge NGS and multi-omics data analysis. And here, from real data set to open source tools, you will build practical skills, earn certifications, and complete a mini project. Now, what I would like to do is to quickly take you through this. What are the various resources and the study materials that will be available to you during the program, and also help you understand what are the various topics that are covered, and also help you get a glimpse into some of the student outcomes that you can expect at the end of the program. Okay, so to do that, I would like to encourage all of you to create an account on the OmicsLogic Learn portal. I'll just stop sharing my screen and reshare it once more so that I can show you the sign up process. So most of you might have an account on the platform, but for those of you who don't, let me share the link. That is where all the resources and the study materials for the program are put together. So if you're interested to explore before enrolling, you can do the same. Okay, so I've shared the link in the chat box. So all you have to do is a very simple process. Visit the link and click on login, and this will redirect you to the sign up page. So if you don't have an account, just click on create an account. You can enter your name, email ID, enter your password, and you can create an account. If you already have any existing social media handles such as Google, Facebook, Apple account, GitHub account, or even Twitter, you can also create an account using any of these options. Okay. Now, for those of you who already have an account, just click on login now, enter your existing email ID, password, and then just click on sign in. Okay, so I'll just pause for a couple of seconds for those who are interested to create an account. I'll just pause for a couple of seconds and wait. And in the meantime, let us know where you're from and what is your background and what are some of the topics that you're looking forward to learn in the program. That will help us also. There's a new recently launched program, and your feedback will definitely help us in incorporating some of these suggestions and also tailoring it to your research requirements and your interest. Okay, so I'll be looking forward to some of these in the chat box, and I hope all the participants are signing up. And in case you're facing any issues while signing up, you can definitely let us know in the chat box, and our team can take a look into it as well. I hope the participants have now logged into the account or is now in the process of logging in. Meanwhile, they're logging in, I'll just briefly go over to the next steps. That is, once you sign in, the very first interface that you see is the courses page, right? Before we explore the courses page, let's first of all explore what all are the various topics that are covered in the program on NGS and multi-omic data analysis. To have a look into it, just head over to this programs here and then just click on training program. Now, once you're on this program page, I'll also share with you the relevant links to the pages. So one is on genomics. So this program, as I mentioned, will be mainly focused on 4 omics type, that is on genomics, transcriptomics, metagenomics, and proteomic data analysis. So the very first omics that we will be covering in the program is on genomic data analysis. Now, let me open it. I'll also paste the link in the chat box. So for those of you who are interested, they can explore it. Okay. So this is the main page for the genomics program. Now, what are the various topics that are covered here, right? So if you're someone who's interested to learn more about a genomic topic and DNA, this program will be ideal for you. So first of all, there will be 2 levels within the program, that is beginner and intermediate. And there are around 12 sessions in total that you will be covering throughout this. So in the first session, what are the various topics that are covered, right? So first of all, it will introduce you to the field of bioinformatics and NGS. We will begin by revisiting one of the greatest scientific milestones, that is the Human Genome Project. You will also get introduced to the evolution of genomic technologies and understand why bioinformatics is the backbone of modern biology. We'll also explore how genomic data is used not just in health research, but also in developing new drugs and improving crop yield in the agriculture. And in the second session, once we have the sequencing data, now we need to organize and clean it. So that is exactly what we will cover in session 2. Here we will break down what are the various common file formats in which we will find, such as the FASTQ, BAM, and BCF. You will also get familiar with key genomic databases, such as the NCBI. We will also explore some of the comparative genomic databases and so on. And we'll also teach you how to extract and prepare such data for further analysis. Okay, and from third session, we will start with some of the hands-on practice. So here we start with the basics of sequence alignment, which is a method to compare DNA or protein sequences. And when it is done across multiple samples, it becomes multiple sequence alignment, which is a key step in understanding how genes evolve and how viruses mutate. And here you will be also introduced to phylogenetics that will help us answer questions like how did 2 species evolve from a common ancestor and how closely related are 2 strains of virus and so on. Okay, and in session 4, which will be a little bit more practical hands-on on phylogenetic, where you will build your own phylogenetic trees, and you will also apply what you have learned to real data set and understand how molecular evolution can be inferred through such data. And from session 5, this session will introduce you to doing all of these analysis using R programming. So if you're someone with a knack for coding, especially this session might be of interest to you. You will learn to import biological sequence data, perform MSA using R packages, and even run phylogenetic analysis using R coding. We'll also cover how DNA strands replicate and how to compute reverse complements, all using R as well. And in session 6, here we get to one of the most popular topics, that is on variant calling. Here we will show you how to align reads to a reference genome and detect changes like SNPs, indels, and structural variants. You'll also learn how to assess the quality of your variants and apply the appropriate filters. Now moving on to the intermediate level. So now here onwards, we will be covering a little bit more complex topics that is different from the beginner level. Here you will run practicing a complete variant calling pipeline, starting from raw sequencing reads and ending with a VCF file of annotated variants. And you will also get hands-on experience with visualization tools like Integrative Genomics Viewer, where you can visualize your mutations on the screen. Now, the next topic is on functional prediction of variants. So now that we have found the variants, the next big question that usually ask us, what do they actually do, right? So in this session, you will learn how scientists predict the function of a variant, whether it disrupts a gene, causes a diseases, or has no effect at all. So we'll also talk about the types of annotation, challenges in interpretation, and new trends in the field. And in session 9 is all about visualizing your data using tools like UCSC Genome Browser and IGV, where you will learn to navigate through across the chromosomes, zoom in on genes, and visualize where these mutations are occurring. And in session 10, that is interpretation of genomic variants, we will learn to classify variants. Is it pathogenic? Is it benign? Or is it uncertain? We will also walk you through the ACMG guidelines for classification, explore genotype phenotype connections, and show how researchers filter large data set to focus on clinically important data. Okay, and in the second last session, that is on genome-wide association studies, which is also one of the topics that is mostly requested by the participants, which is a powerful technique used to basically study how genetic differences relate to traits across large populations. Here you will explore everything from the study design, SMP, genotyping, data analysis to polygenic risk scores. We'll also touch a little bit on the ethics of using genomic data at such a large scale and why GWAS is both promising and is also a little bit complex. And finally, we will culminate in the last session that is on research specialization areas. So here we will explore where genomic data analysis is making the biggest impact in today's research. You'll be introduced to different fields like cancer genomics, infectious disease genomics, population genomics, and so on. Okay, so these are the main topics that are covered under the genomics. So now, if you are interested to learn or you have some more topics that you're curious to learn more about, or maybe some topics that you're interested, you can also definitely put it in the chat box and we'll be happy to understand that. Okay, so that is all about the genomics. Now, the next topic that will be covered is, as I mentioned, on transcriptomics. So let's take a look at what all are the topics that are covered here. I'll share the link in the chat box once again. Okay. So this is the program for this is the module on transcriptomics. And in session 1, we will start at the very beginning with transcription and the central dogma of molecular biology. We often have participants who are not from a biological background, so that is why we have incorporated very basic level of biology as well. And we'll also explore the evolution of sequencing technologies from the very famous classic Sanger sequencing to today's high-speed platforms like Illumina, Nanopore, PacBio, and 10x Genomics. And we'll also cover some sequencing types like targeted sequencing, whole genome sequencing, whole exome sequencing, and how transcriptomics data is collected through various technologies as well. And now in session 2, that is on sources for data extraction, you will learn to explore how to extract such data from public databases like TCGA, there is CCLE, GDSE, Firebrowse, NCBI GEO, Bioproject. These are several of some of the databases that we are covering in this module. And here you will also practice extracting such real data from these data repositories, also understanding what are the various file formats in which you can find them, and also preparing them for further analysis. Now, in session 3, you will learn the full pipeline, that is right from the logical aspects of pre-processing, mapping reads to reference genome, and quantifying gene expression levels. We'll also talk about why cleaning your data matters, and you will also be introduced to the key concepts like normalization, log transformation, quality checks, and so on. Okay. And now moving on to session 4, that is on exploratory data analysis. So here we will be covering techniques like PCA, TSNE, MDS, and simple pivot tables to see the patterns in the data that you're analyzing. We'll also introduce basic statistical tests like t-test, ANOVA, FDR, and even show how to run differential gene expression using nothing more than a simple Microsoft Excel. Okay. And now in session 5, now that we have identified the differentially expressed genes, the next question that you will be asking is, what do these genes actually do? right? So here you will explore pathway databases, learn to annotate your results, and also identify biological functions behind your data. We will guide you through real hands-on pathway annotation so that you're able to connect the gene changes to diseases, cell types, and the biological processes. And the final session of this level, that is on pathway annotation. So here we will zoom out and ask question like which group of genes are working together. And to resolve this, we will be using tools like gene set enrichment analysis and explore gene ontology terms like biological processes, molecular functions, and cellular components. We'll also navigate the KEGG pathway, mapping your data to larger biological systems. Now let's take a look at what are some of the topics in the next level. Here we will be covering a little bit more complex level of analysis aspect. So the first here is on pathway annotation. And here we will go beyond just the list of genes that we covered in the previous level. Here you will be using tools like N-symbol, G-profiler, and string to understand how such genes are interacting at the protein level. And in session 8, here we will focus entirely on visualizing the results using something called cytoscape. And here you will build network graphs to show gene relationships, functional pathways, and clusters of activity, all in an intuitive and visual way. And the next session is on machine learning. Again, one of the very interesting topic for several of the participants. You will try to understand both the unsupervised techniques like clustering, k-means, and hierarchical, and supervised methods like decision trees, random forest, SVM, and LDA in the context of RNA-seq data. And we'll also teach you about feature selection, how to find the most informative genes from thousands of options that are there. Okay. And then now, if you're new to coding, no need to worry at all. We'll be starting with the very basics of R in session 4, that is on getting started with R for RNA-seq data analysis. You will learn to use R for loading and transforming expression data and creating plots like box plot, heat maps, scatter plots, and histograms, and so on. And finally, in the last section of this level, that is on exploratory data analysis, we'll wrap things up with advanced visualization and dimensionality reduction in R. You will generate and customize PCA and TSNE plots, color them by clusters or by condition, and interpret what those plots reveal about your data. So these are some of the topics that are covered in the transcriptomics module. Okay. Now, I guess participants have not responded in the chat box, but please feel free to share any questions that you might have about the topics that are being covered, or if you are interested to learn about any particular topic in depth, you can also mention that, and we'll be happy to take it up. That is about the transcriptomics. Now, the third module that is being covered is on metagenomics. So here, let's take a look at what are the various topics covered here. And I'll share the link in the chat box. Okay, so here in session 1, we will be basically exploring what exactly is metagenomics and why it has become such a popular term in everything from health to environmental science in biomedical research. So this session basically lays the foundation by exploring how metagenomic compares to other omics technologies. And in session 2, once we know what we want to study, the next question is how do we capture that information? So this session basically demystifies the core sequencing approaches like shotgun sequencing, 16S rRNA, amplicon, and targeted gene sequencing. I guess these are some of the terms that a lot of the participants might be familiar with if you're someone who's interested to learn about metagenomics. Now, before we analyze, we must prepare. That is basically what we will cover in session 3. But what makes a high quality metagenomic data set, right? So how do we identify it? So from DNA extraction methods to library preparation and quality control, in this session, we will walk you through the key steps that is needed to ensure reliable, clean input for downstream data analysis. And in session 4, you will learn to navigate open source public repositories like NCBI, SRA, and HMP databases. How do you identify the right data set for your research question? And what are the ethical considerations of working with clinical versus non-clinical data? These are also some of the topics that we will cover here. Now, as you might know, raw data are often messy. So how do we clean and organize them? That is what session 5, that is on tools for metagenomic data processing, is all about. Here we will dive into pre-processing techniques such as quality filtering, adaptive trimming, and then move into assembly and binning. And in session 6, once you have processed your data, how do you begin to understand what's there and what they are doing? So, this session basically covers key concepts like taxonomic profiling, species identification, and functional annotation using gene prediction tools. Okay. Now, moving on to the next level, that is on intermediate, let's explore what are some of the topics that are covered. Now, here on session 1, that is under intermediate level, here we will zoom into a more refined look at the microbial diversity using the data to pipeline. And here we will try to answer some of the questions like how do we extract and prepare the data for this analysis? What can amplicon sequencing reveal about the subtle shifts in the microbial composition and so on? And in session 8, now that we have understood the basic pipeline logic, but next what happens? From alpha and beta diversity plots to OTU heat maps and abundance table, in this session, we will teach you how to interpret what the data is trying to tell you. And in the next session, that is on kind to pipeline, which is another powerful toolkit. But how does it compare to data 2, right? And here we will explore the complete workflow from pre-processing and demultiplexing to taxonomy classification using referent databases. And in session 10, that is on diversity analysis and result visualization in kind 2. Here we will understand microbial communities. Understanding microbial communities means comparing them. So in this session, we will walk you through alpha and beta diversity metrics and how to visualize these patterns in a meaningful way. And finally, the session will conclude this with what does all of this look in action. So here we will hear from experts who are applying metagenomics in unique contexts, such as femtech and maternal health. And these perspectives will help you connect the dots between methodology and impact, and that is highlighting the relevance of what you have learned so far. Okay, so that is what is covered under metagenomics. And the final module, that is on proteomics. Let's take a look at what are the topics covered here. Okay, I've shared the link in the chat box. Okay, so here we'll try to understand what is proteomics and how does it connect with genomics and transcriptomics. And in this opening session, we will also explore where proteomics fits into the omics overall hierarchy and why the protein layer is often where function, disease, and drug targets come to life. And in session 2, we have been studying proteins for decades using techniques like Western blotting, ELISA, and IHC. But are these tools enough for the modern science? So that is basically what we will cover here, where we will walk through the limitations of traditional antibody-based methods and how mass spectrometry changed the entire research. And you will also discover how the Human Proteome Project took on the massive task of mapping all the proteins and why missing proteins are still a mystery that is waiting to be resolved today. And now in session 3, we will ask the question like, how do we go from protein measurement to predicting disease? So in this session, we will dive into biomarker discovery and the role of omics in understanding conditions like COVID-19. And what makes a good biomarker? How can machine learning make sense of thousands of data points to predict the disease? So all of these topics are also some that we cover in the session. And in session 4, what exactly does mass spectrometry identify proteins? And this session also introduces the core technology behind modern proteomics, such as what are TOF, Orbitrap, and ion trap analyzers, and why does it matter, which one you use, and you will also learn how mass spectrometry transforms fragmented peptides into precise protein fingerprints. Now, in session 5, we will explore the advanced mass spectrometry strategies like top-down versus bottom-up proteomics and MSM1 versus MS2 scans. And why do we fragment peptides? What's the difference between DDA and DIA? And how do we quantify proteins with labels or without? And this session is all about how they affect your results as well. And finally, in the session 6 of this level, once you have the raw MS data, what comes next? So you'll get hands-on with Mascot, one of the most widely used tools for identifying peptides and proteins that are used by researchers even today. Now you will learn how to set parameters like modifications and FDR, and you also learn how to interpret the output that's as complex as the proteins that you're trying to study. And in the next level, let's explore what are the topics covered here. And before you diving into the discovery, we need to clean the data. So, how do you handle missing values, normalize the measurements, and spot outliers? So, this session will practically give you the tools to improve the data quality through statistical testing, PCA, and batch effect correction, because dirty data can often mess up with the results as well. Now, once in session 8, that is biomarker identification and statistical analysis, here we will walk you through using meta-analysis to perform transformations, generate PCA plots, and conduct hypothesis testing. And what's the role of FDR and confidence intervals? How do we use tools like PLSDA to pinpoint potential biomarkers? So, this session basically sharpens your ability to find patterns in the proteomics data that you're working with. And in session 9, what's in a raw data file and how do we make sense of it? And this session will also take you inside the mass spec file formats and walk you through workflows using FragPipe. You will also learn how peptide and protein identification really works, from chromatograms to retention times, and how LFQ and match between runs enhance the quantification. Once you have got your differentially expressed proteins, what do they mean biologically? This session teaches you to map proteins to pathways using KEGG and reactome, some of these topics that we have previously covered in the others as well, using visualized networks, using string and stitch, and connect your findings to biological function, key regulators, and even potential drug targets. And in the second last lesson, we'll try to answer questions like, why is it so hard for the immune system to recognize and fight cancer? And in this session, we'll also explore how cancer evades immunity and the role of checkpoint inhibitors. We'll also cover topics on CAR T cell therapy, its mechanism, and why relapse still remains a major challenge in today's biomedical research. And finally, in the last session, that is on proteomics for cancer target discovery and therapy development, we will try to answer questions like why stop at genes when proteins are the main targets of most drugs. And this session basically brings it all together, showing proteomic data feeds into how proteomics data feeds into cancer immunotherapy development. And from identifying targets to validating them, you'll also learn how mass spectrometry and multi-omics integration are shaping the next generation of therapies that we are developing. Okay. So these were the four major modules that will be covered in the program on NGS and multi-omic data analysis. But apart from that, we also receive a lot of questions from the participants who might be interested to learn other topics like structural biology and also learning how to use GitHub. So we also cover a lot of those through the course library that we have. To access it, just head over to this courses tab here and then just click on course library. And I'll paste this. So once you sign in, this was the very first page you must have seen. So this is practically the place where a lot of the coursework and the technology-based coursework and the application-based example projects are covered here. So here you can explore some of the other topics that you might be interested to explore, like structural biology. Yeah. Oh, sorry, let me sign in. So these are some of the other coursework that we have. And as I mentioned in the lessons in these slides as well, let me share it in the chat box, the link, if you're someone who's interested to explore. So all of these coursework, they are fully automated into comprehensive lessons, and there are detailed instructions. There are also short tutorials within the lessons. There are quizzes, there are assignments to test your knowledge, and so on. And all of these also include hands-on practicals as well, so that you don't just miss out on the topics. So this is the first few lessons will definitely be open. So if you're interested to explore what the lessons looks like, you can just open up and just quickly explore it. And another topic that is most asked by participants or students are about GitHub. What is GitHub? How can we learn how to use GitHub for bioinformatics? So this is also one of the coursework that you'll be getting access to and that you can explore. Let me share it in the chat box. Okay. So this is also a very comprehensive lesson. It will walk you through right from beginner level and then slowly build the skills up to practical use of GitHub. So as you can see, there are around 10 lessons and it is very comprehensive. So that is through the coursework, asynchronous coursework, that you can still cover some of these topics that I mentioned. And apart from that, when you enroll in the program, you also get the opportunity to enroll in any one of the workshops that we conduct in a month. So to explore those, here under the programs tab, if you head over to the workshop, this will redirect you to the various workshops that we have. So here there are some of the asynchronous versions of the workshops, and we also conduct workshops live. And there are multiple workshops that we conduct in a month. So when you enroll in the main program, you automatically get the option to choose any of the workshop that you want to attend in that particular month. So it could be depending on the topic of your choice. And just to give you a brief overview of what all of these topics are. So if you're someone from a plant biotechnology background, there are workshop on RNA-seq data analysis for plant biotechnology using R coding, or let's say you are someone who's interested to learn coding, right? Very core basics of coding. And we also have several workshops on coding as well here in this way. And let's say you're interested to learn Python. There are workshops on Python as well. And this is the full list of workshops that we have on several other topics like cancer genomics, metabolomics. If you're someone who's interested to explore the bioinformatic applications in immunology, there are workshops on immunoinformatics as well. Okay, so that is just a quick overview that I wanted to give about the workshop aspect as well. Now, the final component of this program is the mini project. Now, before we get to that, a lot of the times participants might have questions about the coursework that they are reviewing, or they are trying to execute a particular code, but it is not working. So for such students, there will be live mentor sessions as well that will be conducted, and it will be held over Zoom. So the participants can join. The participants will be notified earlier itself, and they can join and ask their questions to the mentors directly and get any queries cleared as well. Okay. And the final component that I wanted to cover was the mini project opportunity under this program. So for participants who might be interested to work on a project under the guidance of a mentor, after covering all of these analysis topics like genomics, metagenomics, transcriptomics, and proteomics, and now if you want to apply all of your learning into a real world data project, then you can do so as well in different specialization topics as well, such as it could be on precision medicine, population health, biotechnology, agriculture, space and astrobiology, and so other topics as well. Now, if you're someone who is curious to know what it looks like or what are some of the outcomes that you can expect, I would like to just quickly walk you through a few examples on how students have completed a research project to achieve their academic and professional goals, but also in a wide variety of ways that they have achieved it. So here on the screen, you can see a cohort of systems medicine graduate program students from Georgetown University who worked on machine learning projects to address various biomedical research challenges. Students also want to complete a research project that they can present at poster presentation competitions and conferences that through which they can win exciting prizes. Now, students might also want to work on a project to get a research publication done, or that gives them this extra competitive edge over other students while applying for higher studies or in the job market. And one of the unique highlights is that faculties and researchers have also worked with us to complete research publication. And if you're someone with a knack for business and science, there is also an option to pursue the entrepreneurship and business innovation track. So this is just the overall view that we wanted to give you about the program that we are recently launching. Now, let me quickly stop sharing my screen. As this is a new program that we are launching, we want to also get a little bit of feedback from you, because of which we have also prepared a short form We have covered a lot of the modules that most of the participants are interested to learn more about. We often get questions from students and even working professionals that they want to learn specific omics data, or they want to collect, they want to understand several omics data types. So, because of which we have launched this program, and we also want to understand from you if there are any feedback that you can give for the program. So, we'll share this link in the chat box. And you can also express any suggestions for improving the program as well, and address any questions that you might have. So just to show you what the form is about, so here you can just enter your basic details. And you can also mention some of the omics that you're interested to learn more about. So some of these we have already mentioned, that is transcriptomics, genomic, metagenomics, proteomics. If there are any other omics that you think that you might be interested to learn more, if there is more popularity around that, omics will be happy to include that in the curriculum as well. So you can just mention that here under the others tab. And what type of project that you want to work for your mini project. If you could mention few details about that, that will be also helpful. And if you could also mention a little bit about your experience working with multi-omics. Are you a beginner? Are you intermediate? Or are you advanced? You can mention that as well. And also, there are also a lot of questions that we get about learning to use specific tools and softwares or platforms for their analysis. So if there are any specific suggestions that is not covered in the curriculum that was discussed today, you can definitely mention that here. And if there are any other general feedback or suggestions that you would like to see in the program, we'll be happy to include that as well. And we will be launching this in the month of July, and it is planned to be a 4-month online program. And we will be opening applications for students to enroll right away. So if you have any questions, you can definitely reach out to our team. I'll just quickly show you the contact details of our team on the screen. So you can just quickly note it down. You can either contact them via phone, or they're also available on WhatsApp. And if there are any questions, you can also email us on communication@omicslogic.com, that is displayed on the screen. So that is just a quick overview that we wanted to give about the program that we are launching. Now, let me just quickly take a look at the chat box in case participants have any questions. Okay, so Rashmi is asking, will we get certificates after completion of the course? Yes, so whenever you complete a course or a workshop or the program, you will be getting a certificate for that. And it will be a digital certificate, so you can add it to your resume or your LinkedIn profile, so you can showcase it. So Surya is asking about the PG diploma program that we had with, okay, so we'll be starting it soon. You'll be definitely notified when we relaunch the program. And in the meantime, if you're interested to explore any of these topics, then you can definitely reach out to us, Surya. And Maria and Rashmi are asking, are these paid courses? Yes, so this is a paid program, and it will be launched in the month of July, but early bird registrations are going on. So for the webinar attendees, there will be definitely discount options available. So you can just fill out the form and let us know your feedback, and our team will reach out to you. So. Okay, Michael is asking how many credit unit. So as of now, we have not specifically mentioned any credit unit, but the duration of the program will be for 4 months. And in this 4 months, all the participants will be going through the training aspect. That is, just to summarize what I mentioned, that is on, you will get trained in genomic data analysis, transcriptomic data analysis, metagenomic data analysis, and finally the proteomic data analysis. And it will culminate in a mini project that you'll be working with the mentor that you can showcase on your resume or on LinkedIn. And do we have any other questions? Okay, so will the classes be daily? Okay, so the lecture videos are already available on the learning online classroom that I just showed you right now. But apart from that, there will be mentor sessions once a week. That will usually might be most likely will be on weekends, where if you have any questions while going through any of these coursework materials and you are getting stuck somewhere, so you can definitely attend these live mentor sessions and get your doubts clear. And other than that, also, you can definitely email us, and the mentors can reply back to any queries that you might have. So Surya is asking how much is the cost of the 4 month program? Yes, so currently the program, we are still in the process of finalizing the curriculum, and that is why we also wanted to understand from the participants what all topics that they want to cover. But yes, our team can definitely reach out to you. Surya, right? Yes, our team can definitely reach out to you and let you know about the cost of the program. Any other questions? Let me share with you the form once again in the chat box. So this session will be is getting recorded right now. So in case you want to go back and revisit any of the topics that was discussed, or let's say you joined the meeting late, you can definitely go back and review it. We will email you the link to the recording for today, and just make sure to keep an eye on your inbox. Once the recording is available, we will notify you. So Ban was asking, after joining the program, are we free to join any workshop within the time period of 4 months? Yes, you can join any one workshop in the 4 months of time. So in total, there will be around four workshops that you can attend. So we'll give you options. There are at least 3 to 4 workshops that we conduct in a month, and you have the flexibility to choose any one from it. And some of the topics that we are conducting workshops are on, like I mentioned, on immunoinformatics. There are some coding workshops that we conduct, like R coding, and we also conduct some specific topics like cancer genomics. If you're someone who's interested to explore some omics type outside of the ones that is being covered, like metabolomics, we also have workshops on that. So you are free to opt for that. Okay, so Akanksha is asking what will be the tentative timings of the program. So the program, the lecture recordings, it will be made available on the online classroom. So even if you're a working professional or let's say you're a student, you can still go through it at the time. But there will be live mentor sessions once a week that will be held on weekends that you can join. So it will be usually during evening hours because we also have a lot of international participants so who want to attend and ask their questions. So it might be usually around like 7 p.m., 7:30 p.m. at IST, around that time. And the program, if you're asking timing days about that, it's probably going to be launched in the month of July, but we will definitely notify you of the exact date via email as well. And Jeffrey is asking, will the program help in topics related to systems biology and aging research? Yes, Jeffrey. So we actually have a plan to launch a workshop on aging and health related research. So, but it is still under the process of planning. So it will be covered under the workshop, but not in the program modules that we are covering. So once that is launched, I think that will be helpful for you to attend. And it will be a live workshop as well. So you'll be attending these workshops, and these workshops will also be conducted during the evening hours so that students and working professionals after your school, college, or work, you can still attend it. And all the recordings will also be made available. So, in case you're not able to attend a specific workshop, there are still recordings that you can go through. And if you have any questions after watching the recording, you can also email us and we can pass on the question to the mentor and they'll help address. There is next question. As a beginner, what better for me start by bioinformatics program or omics program? So this program that is on NGS and multi-omic data analysis is going to be completely beginner friendly. So it's not going to be a problem. We'll start right from the basics and then help build each of the concepts one by one. Kavita, will it be covering only beginner level or intermediate level? Also for courses described under 4 month of program. So we'll be covering till the intermediate levels. So in total, there should be around 12 sessions for each of the modules on genomics, transcriptomics, metagenomics, and proteomics. Then a next question. Surya is asking, video recordings will be provided. Yes, so the lecture videos are already made available on the platform, so you can still go through it at your own end as well. Will there be placement assistance, ma'am, like PGD program? So there will be assistance in terms of the recommendation letter that the mentor will provide. If you want to work on a research, let's say you're working on the mini project that you want to further develop into a project, there are also possible opportunities for that as well to get a publication done, things like that. So that will be an additional add-on to the existing program. But if you have any specific questions like that, you can definitely connect with our team. They'll be able to walk you through step by step how you can go through that. Any other questions? We'll also email you once the recording is available. We'll also email you all of these program topics that are being covered so that you can also have a look at what are the curriculums, what are the tools and technologies that are covered, and details like that so that you can go over it at one go. So Jeffrey is asking, what will be the payment channels? Because I'm in Nigeria. Um, so for international payments, I guess the sales team will be able to assist you. Jeffrey, can you maybe share your email ID in the chat box, and I can let the sales team assist you. But there are different payment channels that are there, like Stripe, PayPal. So one of these will be the option for you. As a beginner, what better for bioinformatics program or omics program? Yes, so the program is completely beginner friendly. Is that the question? As a beginner, what? Yeah, so we are covering both elements of that. So you're asking, I've already registered for the PG diploma program and I've registered again when the course start. Okay, so that will be a different program that we will be conducting. I think we'll be relaunching in a couple of months later, and it is still under review and a little bit more updating of the curriculum and some of the topics. So once that is launched, we'll definitely let you know. So this will be a separate program from that. It will be a little bit more of shorter duration. The PG diploma was 1 year duration, and this is a 4 month program. Any other questions? Okay, I think that will be all. There are no further questions, but in case you have any specific questions that you could not ask today and would like to get maybe a little bit more guidance on, you can definitely reach out to us on the contact details that we have shown in the screen. So we'll also email you the contact details so you can get in touch with us, and you can also email us. So somebody asked about payment and cost. Okay, yes, we will be sharing with you a detailed information about the payment and cost details and also the payment options that are available for the participants. Okay, then. Thank you so much, everyone, for joining. And we look forward to seeing you all in the program that will launch in July. There is still time. So if you have any questions, you can definitely reach out. So Hussein is asking, is there any attendance certificate available? No, Hussein. This is just a program launching webinar that we are conducting. Certificates are available for the coursework that you can go through or any of the programs for which you will get. Okay, perfect. Thank you so much, everyone, and we look forward to seeing you all in the